Let's understand photorespiration. In case of photorespiration, we saw Kelvin cycle. Now in Kelvin cycle, we saw that the main process was of carbon fixation. All right. Whereas in this process, we would find that it is, instead of carbon fixation, carbon is given out. But this process actually prevents 75% of carbon from losing, but loses 25% of carbon. Now let's understand how does the process occur. Now in this process, there are three organelles which are involved, and these organelles are chloroplast, peroxisome, and mitochondria. Alright, now let's start with chloroplast. In chloroplast, what happens is RUBP, ribulose biophosphate, which we understood in Kelvin cycle, gets oxidized and forms phosphoglycolate and PGA. We know that this is 5 carbon molecule. That 5 carbon molecule splits up into 2 carbon and 3 carbon molecule. 3 carbon molecule is PGA. PGA used in for Kelvin cycle. PGA, this is the structure of PGA. We are well versed now, as I had explained it in my earlier video. And phosphoglycolate. To understand phosphoglycolate, let's understand this structure. This is the structure of glycolic acid. Glycolic acid is CH2OH, COOH. Now this OH gets phosphorylated to form phosphoglycolate. Now this phosphoglycolate gets hydrolyzed. Inorganic phosphate is removed. So this inorganic phosphate is removed, and what we end up is with glycolate. Once it forms glycolate, it leaves chloroplast and moves into peroxisome. Now what happens in peroxisome? Glycolate oxidase. As the name suggests, whenever ACE is there, that itself means it is enzyme, and enzymes we know are biological catalysts. So glycolate oxidase oxidizes glycolate to form glyoxylate. Now this CH2OH which was there got oxidized to CHO. So oxidation occurs. Glyoxylate is then acted upon by glyoxylate aminotransferase enzyme and converts glyoxylate to glycine. Now glycine is amino acid. Now why amino acid? Because it has amino group and carboxylic group, so amino acid. And we find that to this carbon NH2 has been added. Alright, so glyoxylate aminotransferase. Why? Because amino group has been transferred to glyoxylate molecule to form glycine. Now once it forms glycine, it leaves peroxisome and gets into mitochondria, the powerhouse. Now here what happens is, this glycine is acted upon by glycine decarboxylase. 2 molecules of glycine are required here. Now when we say 2 molecules of carbon, that means total number of carbon gets to 4. 2 carbon of one glycine, 2 carbon of another glycine. So these 2 carbons. Alright. Now glycine decarboxylase enzyme acts on it and strips off ammonia and 1 carbon dioxide. So ammonia is also lost to environment and carbon dioxide is also lost to environment. So 1 carbon is lost, that is 25% carbon is lost from the system. So in this step, we got rid of NH2 as ammonia and one of the carbon as carbon dioxide. So 3 carbon molecule is formed, and that 3 carbon molecule is serine. 1, 2, 3. 3 carbon molecule is serine, and from one glycine, NH2 is lost in form of ammonia. So, and one NH2 is left, which is here. So this is what is serine, yet another amino acid. Once it forms serine, it quits mitochondria and enters peroxisome. We know peroxisome. It's meant for oxidation. So what we find is COH is intact, CH2OH is also intact, but here it gets oxidized to carbonyl, C double bond O. And this molecule, we call it as hydroxypyruvate. We call it as hydroxypyruvate. Alright. When the molecule moves in from hydroxypyruvate to glycerate, what happens is it is reductase enzyme which works on. Reductase obviously means reduction, hydrogenation, and NADH is being used where hydrogen is utilized and NADH gets converted to NAD+. And this hydrogen is used for what? This hydrogen is used for hydrogenation of this carbonyl, C double bond O. So this carbonyl, the one which is in the center, gets converted to CHOH, whereas COH remains intact and CH2OH also remains intact. Now this is what we call as glycerate. Alright, now once it forms glycerate, then it has to quit peroxisome and get into chloroplast again. Alright, now in chloroplast, it forms PGA, phosphoglyceric acid. Phosphoglyceric acid, the structure which I have already mentioned over here, it is that OH which gets phosphorylated, owing to which one ATP is being used to form ADP. So we get PGA. So we are back again here, and then with RUBP, the structure I have mentioned over here, the cycle keeps on happening. The entire process requires bright light, high temperature, and high oxygen and low carbon dioxide. And this process is also known as PCO, photosynthetic carbon oxidation cycle, PCO. Alright, and obviously this process is not for fixation. Yes, it does protect 75% of carbon, but loses 25% of carbon as carbon dioxide. That's what I have for photorespiration.